Good morning, everybody. I am honored to continue today's presentations, and my talk will be related to vascular effects during neuroinflammation. Everybody knows this, what is neurovascular unit and how it works and all that, but what if effects are coming from vasculature to neurons? In that case, there is a suggestion, and not only by me, I guess Logovich also proposed the same thing, to call this unit the vascular neuronal unit. Little bit about fibrinogen. That is the component of plasma that is synthesized in hepatocytes. Therefore, it is only in blood, nowhere else. And any effect that comes from fibrinogen, this would be coming from vasculature. When fibrinogen concentration is increased, that phenomena is called hyperfibrinogenemia. During inflammation, you know that C-reactive protein, which is a very good marker of inflammation, shoots up, increases quite high, but when inflammation subsides, it decreases drastically and very fast. While fibrinogen, as you can see, increases slowly and stays elevated for about 20-21 days. And again, if you can see, concentration of fibrinogen never goes that high as, for example, for C-reactive protein. It just doubles, and that doubling of concentration is enough to cause all the problems it can cause. It is increased during hypertension, diabetes, stroke, causes pulmonary disorders. It hinders immune surveillance of tumors. It affects lungs, affects peripheral circulation. These are the effects, among others, which I just presented to you, of effects of fibrinogen. We tested the effect of fibrinogen on permeability, and we found that fibrinogen induced increased crossing of albumin through endothelial cell monolayer, and this effect was dose-dependent, as you can see. Fibrinogen itself can cross this endothelial cell monolayer, and again, it is dose-dependent effect. We developed a method to define vascular permeability, whether it is a transcellular or paracellular. So we used dual probing method. So we used two probes, which is one is fluorescence, and it's called fluor here in this slide, and the bovine serum albumin, which is conjugated with Alexa 647. We compared crossing of these dyes through vasculature. And as you can see, during inflammation, which is hyperfibrinogenemia, when gaps are opening, and you know that gaps are always opening and closing with various frequencies and various amplitude. So if inflammation is high, they're opening more. And that's what we see here. But then after some time, the gaps are closing. While gaps are closing, at that time, movement of bovine serum albumin continues. That tells us that permeability of vasculature is mainly through transcellular transport. Then we tested this, whether this effect was caveoli related, and yes, we found it that it is through caveolar transcytosis, because when we use the C-reactive cRNA against caveolin 1, we blocked it, this effect, so we decreased caveoli 1. We saw this transcellular transport has been decreased. This is the mechanism of all this. This is the transcellular pathway that involves caveoli and plasma level vesicle associated protein, which are forming caveoli membrane. And this is paracellular pathway. So fibrinogen binds to its receptors on endothelial cells, ICAM1 and alpha-5 beta-1, activates ARC signaling, and that causes activation of caveolar movement. At the same time, fibrinogen, hyperfibrinogenemia at high level of fibrinogen, it activates mitochondria. Bedaquiline decreases fibrinogen-induced transcytosis. That tells us that if we remove ATP, we affect mainly transcytosis rather than paracellular transport. Again, we used dual probing method. Here we tested whether this hyperfibrinogenemia induces caveolar formation of ATP, and yes, it does. So when fibrinogen is binding endothelial cells, it decreases. We have, as a result, decrease for ATP. That means ATP has been used. And then if we use myoricin, which is blocker of sphingolipid synthesis, we see that caveolar transcytosis is affected. So that again proves that use of ATP relates to caveolar transcytosis. As a neuroinflammatory disease, we used traumatic brain injury, and particularly method of cortical contusion injury was used. And we used mild to moderate TBI, as this is the case when we have mainly memory being affected, and it is a lasting effect, while, for example, severe traumatic injury does not affect short-term memory, at least not that much. We proved that fibrinogen concentration increases during CCI or after CCI. This is the place where to show where we study our effects, at least 200 micrometers away of necrotic area. And that's what we see that in about after 1 hour, we see the PL vascular permeability is increased during or after CCI. This is the area, this is the cut, what we do to do the immunohistochemistry. And if this is the injured area, that's the place where we study our effects, at least 200 micrometers away from the injured area. We tested expression of plasma of prion protein and its colocalization with fibrinogen, because we suspected that this is the protein that can be bound to fibrinogen. And as it is on some level of the surfaces of molecule of cells, neurons or astrocytes, this prion protein becomes cellular prion protein becomes a receptor for fibrinogen on these cells. This is the effect of traumatic brain injury on memory, short-term memory, and of course it decreases. Again, if we used cRNA against caveolin-1, affecting or targeting caveolar transcytosis, we see the memory is changed or altered or restored if we block caveolin-1. So if we block caveolar transcytosis, memory during TBI comes back. Not completely, of course. And again, myoricin being used again to target the formation of caveoli. And we saw that it mainly affects transcytosis, transcellular transcytosis, and has no effect on movement of low molecular weight protein, which is a marker for paracellular transcytosis. Here you can see this is a fibrinogen outside of the endothelial cell, and this is inside the alveoli. It is faint, but it's still possible to see that it is encapsulated inside the alveoli. These are conglomerates of fibrinogen, and this is one. So that's how this fibrinogen is taken up by alveoli, moves through the cell, and goes outside in the basal side. What does it do to astrocytes when it goes out of the vessel? We see activation of astrocytes during CCI. Here, let's consider this area, and if we see that there is a colocalization of fibrinogen with astrocytes. Astrocyte is depicted with GFAP. And here you can definitely, clearly see that fibrinogen located outside of the vessel, which is blue, and between the vessel and astrocyte end feet. This red is clearly alone, sitting between the green and the blue. So fibrinogen comes out of the vessel and deposits in between the vessel and astrocyte end feet. It causes increase in ROS in astrocytes, causes neurodegeneration as a result. And this is the depiction of some mechanisms of that. So we have TBI that causes hyperfibrinogenemia, increases transcellular transport, then comes out, binds to cellular prime protein, forms the complex, which is very difficult to digest by any digestive systems, for example, plasminogen or plasmin, and then it sits in between the vessel and astrocyte and causes short-term memory changes. And what does it do to neurons? We see that fibrinogen colocalizes with neurons also, so it can bind to neurons apparently. And we tested that, these effects. So we see that NF-kappa B has been increased, translocated in neurons, so it affects neuronal NF-kappa B formation. Here are more data, and we used ICAM1 and PRPC as blockers to show that definitely fibrinogen binding to neurons is the mechanism that affects this process in neurons. Here again, we see the NF-kappa B being affected and involved in this process. And this is during CCI. While the previous studies were done on cells alone, this is already in a mouse that had been, where we had TBI induced. Fibrinogen also causes increase in CCL2, which is a marker of inflammation, as you know, and interleukin-6. It causes neuronal death. And if we block PRPC or the ICAM, we see that this effect can be changed or altered. Here, using proximity ligation assay, we confirmed that fibrinogen definitely binds neuronal ICAM1 and PRPC. So these are receptors of fibrinogen on neuronal cells, specific receptors. It causes mitochondrial superoxide production on neurons. And here are some conclusions of the study. So at high level, fibrinogen, and this is a vascular effect, that it comes only from vasculature. There is no fibrinogen anywhere found in our body. I'm talking about fibrinogen, not the fibrinogen separate chains, which lately have been found in generated even in brain cells. But there is no indication that fibrinogen is synthesized in those cells. It is synthesized only in liver, and therefore it is only in vasculature. So this is a vascular effect. So it extravasates for cerebral vessels into extravascular space, via alveolar transcytosis. It interacts and activates astrocytes, which affects neurons through PRPC and ICAM receptors, causes formation of reactive oxygen species, overexpression of interleukin-6 and of kappa B, and thus results in neuronal death, neurodegeneration, and all that leads to memory changes. I would like to acknowledge my very helpful coworkers, co-investigators, help from NIH, American Heart, France. And I would like to thank you for your attention. And if anybody is interested to contribute to this special issue, I'll be more than happy. Thank you very much.